Hey everyone, Dr. Mike here. Today, we're diving into a part of your immune system that works tirelessly behind the scenes. It's called the complement system, a group of plasma proteins that enhance immune responses. Think of it as your body's 24/7 security team. A network of over 30 proteins mostly made in the liver that circulate in blood in an inactive state. They wait for a trigger to spring into action. This is innate immunity, ready at birth, acting fast. It complements other immune cells, opsonization, inflammation, and cell lysis. The system works as a fast proteolytic cascade, like a line of dominoes. A small trigger becomes a massive response through amplification. Understanding this is vital for medical students. It also clears old and damaged cells and immune complexes. So how does the cascade get started? Let's break it down. There isn't just one switch. There are three pathways that trigger complement. Classical, lectin, and alternative all lead to the same goal. First, the classical pathway, triggered by an antigen-antibody complex, IgG, IgM. The antibody complex recruits C1, kicking off the cascade. Next, the lectin pathway. Mannose-binding lectin recognizes microbial sugars, no antibodies required. Lectins bind mannose on bacteria, fungi, and many viruses, then activate enzymes. Finally, the alternative pathway. Think constant surveillance, it's always ticking over at low levels. C3 spontaneously breaks down into fragments. Nearby pathogens allow C3b to stick and amplify the response. Bound C3b resists inactivation and triggers positive feedback, often activating early. Takeaway: different triggers, same aim. They converge at C3, producing C3 convertase. C3 convertase is the central hub that drives downstream effects. Once C3 convertase forms, massive amplification follows. It's like a factory worker cleaving many C3 proteins. C3 is split into C3a and C3b. C3b coats pathogens, opsonization, tagging them for phagocytes. Phagocytes recognize the eat me signal and remove invaders. C3a acts as an inflammatory mediator, sounding the alarm. Some C3b joins the convertase, transforming it into C5 convertase. Now the enzyme targets C5. C5 is cleaved into C5a and C5b. C5a is a potent chemoattractant, recruiting neutrophils and amplifying inflammation. Cells converge on the infection site to fight the invader. C5b initiates the terminal pathway, the membrane attack complex. C6, C7 and C8 sequentially join the complex. Multiple C9 molecules polymerize to form a hollow tube, the pore. The MAC inserts into the membrane, causing ion water influx and cell lysis. Complement is a powerful double-edged sword and needs tight regulation. Genetic deficiencies in complement components cause specific clinical problems. Deficiency of C1 inhibitor, a brake on the classical pathway, leads to hereditary angioedema. Episodes of severe swelling of skin, airways, and gut due to unchecked fluid leakage. C3 deficiency is devastating. All three pathways converge at C3. Patients get recurrent infections with encapsulated organisms like Streptococcus pneumonia and Haemophilus influenzae. Without opsonization by C3b, phagocytes struggle to clear these pathogens. Deficiencies in terminal components, C5, C9, prevent MAC formation. This specifically increases susceptibility to Neisseria infections, including meningococcal disease. Complement bridges innate and adaptive immunity, enhancing antigen presentation and B T cell activation. Opsonization improves uptake by antigen-presenting cells and supports adaptive responses. Complement helps clear immune complexes and prevent immune complex disease. Thus, the complement system amplifies defense, links innate and adaptive immunity, and is vital for host protection. Stay safe and be well. If you want, I can also make a neat flowchart diagram to use while explaining.